Almost 60 Australians are diagnosed with breast cancer every day. More than 3,000 will die from the disease each year. But the picture would be even worse had it not been for new methods of diagnosis and treatment.
I use bioinformatics methods to identify the cell of origin for the most invasive form of breast cancer.
Professor Gordon Smyth is a statistician who specialises in genomic sequences. His mission is to understand what goes wrong inside cells when they become cancerous.
So I collaborate with biologists working on different cancers and especially on breast cancer. We were able to identify the cell from the type of breast cancer with the worst outcome, and it was an unexpected cell type, not what people expected. If we can identify the cell that cancers arise from, then we know a lot more about how we might be able to treat them.
Professor Smyth started his career as a mathematician, but moved into medical research 20 years ago, around the same time the human genome was published.
That development of genomic technologies has just continued ever more rapidly over the last 20 years, and it generates ever larger and larger data sets. So now bioinformatics and data analysis are becoming an ever larger part of medical research.
He now collaborates with researchers around the world on cancer and blood disorders, as well as malaria, HIV and COVID. His research was used in the development of venetoclax to treat leukemia.
New genomic technologies are just developing at a fantastic rate, and they also allow us to attack diseases in ways that we never were before. It's really moving to listen to survivors talk about their experiences and to know that without the drugs or interventions that were developed, they wouldn't have been alive.
In recognition of his significant contribution to medical research, Professor Smyth has been elected a Fellow of the Australian Academy of Science.